In Chapter 23, we talked about how acetyl-CoA formed from each of our major biomolecules is metabolized for energy in the citric acid cycle and the electron transport chain. In this chapter, we talk about how carbs enter the body, get converted to acetyl-CoA, are used for anaerobic energy, and are stored for later. We'll also talk about the role of hormones on directing metabolic pathways. We see the same steps we had talked about in Chapter 23, now laid out specifically to carbohydrates. Carbohydrates are digested all the way down to monosaccharides, which can then be converted to acetyl-CoA through glycolysis. Glycolysis and other pathways involving glucose will be the subject of the majority of this chapter, but first, how do we get everything into the monosaccharide form? When we have monosaccharides, it doesn't take much to digest them. On the other hand, complex carbohydrates need to be broken down to monosaccharides. This process starts in our mouth, where the enzyme alpha amylase breaks it down. Alpha amylase, as an enzyme that is optimized for the more neutral pH of saliva, denatures in the acid of the stomach, but is re-released in the small intestine to further break down the remaining polysaccharide chains. Any disaccharides that remain at the surface of the intestinal cells are broken down by specific hydrolases: lactase for lactose, maltase for maltose, and sucrase for sucrose. Only monosaccharides are taken into the cells and then the bloodstream, and mainly glucose, but also fructose and galactose. This diagram shows the path we just described, some digestion in the mouth by alpha amylase, and continued later in the intestines. The intestines are filled with villi, tiny little fingers which give a much larger surface area for the absorption of nutrients. Once we have those monosaccharides in our bloodstream, they can be absorbed into any cells and used in a variety of ways. One of the main ways these monosaccharides are processed is by conversion to acetyl-CoA to be used in our central metabolism. So that's where we're heading. Most of the monosaccharides come in the form of glucose, which is broken down through a series of steps called glycolysis. Glycolysis has two phases: a stage with all 6 carbons of glucose present, and a second stage where the 6-carbon molecule is broken down into 2 3-carbon pieces. The first stage is shown here, where 2 ATP are invested. Notice this step is costing us energy for a process that we ultimately want to produce energy. The phosphates installed in this phase will be made higher energy in the 3-carbon phase and recovered as ATP. In the 3-carbon phase, we can see the actual production of energy. Notice that everything is produced in sets of 2 here. That's because we now have 2 3-carbon molecules that are being processed for each glucose that came in. We get 2 NADH and 4 ATP in the 3-carbon phase, all in the cytosol. But don't forget that we spent 2 ATP in the 6-carbon phase, so that our net yield is actually just 2 ATP and 2 NADH in the cytosol. This cytosol piece is important because NADH in the cytosol can't be oxidized in the electron transport chain directly. Let's put a pin in that and talk glycolysis first. The first step of glycolysis invests in ATP. That ATP can be gained later, but it also functions as a mean to trap glucose inside of a cell. After the glucose is trapped with a phosphate, we isomerize the glucose to a fructose, just moving the ketone over. It doesn't give us any energy, and we don't spend any energy, but it does posture us for gaining more energy later by breaking the molecule down. We can lock us into the phosphate form by adding another phosphate on the other side of the 6-carbon molecule, which helps us later. For now, it costs us an ATP for a total of 2 ATP spent during the 6-carbon stage. As we move into the 3-carbon phase, we don't split the molecule evenly. We need both pieces to be glyceraldehyde 3-phosphate, so the piece that is not glyceraldehyde 3-phosphate needs to be isomerized. You've seen both glyceraldehyde and dihydroxyacetone before. They're the simplest trioses, the 2 that you've memorized. In the 3-carbon phase, we start with glyceraldehyde 3-phosphate. We have 2 glyceraldehyde 3-phosphates for every glucose, one from glyceraldehyde phosphate produced directly and another isomerized from dihydroxyacetone phosphate. With this glyceraldehyde 3-phosphate, we can oxidize and gain energy. The oxidation stores energy in NADH, but also in a new phosphoester bond that can be hydrolyzed later for energy. When we phosphorylate ADP into ATP later, we'll gain that energy back. The NADH is in the cytosol, not in the mitochondria, so we can't think of it in exactly the same way that we did in chapter 23. We'll talk more about that later. Notice that since we have 2 glyceraldehyde 3-phosphates, we get 2 NADHs. Everything is doubled from this point forward. The phosphate that we trapped in the last step is hydrolyzed to make ATP. Again, this step happens twice since we have 2 3-carbon molecules. We get 2 ATP total at this step, but that just cancels out the 2 ATP we spent earlier. We still have the 2 NADH in the cytosol, but really that's the only thing that's our net production at this point. Step 8 is really just an isomerization, but it makes it much easier to get energy out of the phosphate that remains later. Step 9 is a dehydration. Again, this doesn't give us any energy directly, but it does make the phosphate much more reactive and easier to put on to an ADP in order to make ATP. In this final step, we finally see the phosphate removed to generate ATP. Again, since we have 2 3-carbon molecules per glucose, we produce 2 ATP per glucose at this step. This brings us to a total of 2 ATP produced per glycolysis, 2 NADH produced by glycolysis in the cytosol, and 2 pyruvates. So our net yield is 2 ATP, 2 NADH in the cytosol, and 2 pyruvate. This slide does 2 things. It shows you on the bottom left the overall reaction of glycolysis and how, as a net reaction, glycolysis takes 2 NAD+ and reduces them to 2 NADH, 2 ADP and converts them to 2 ATP, and takes a glucose and converts it to 2 pyruvates. It also shows us that while glucose is the major monosaccharide processed for energy through this pathway, galactose and fructose can also be processed through many of the same enzymes for the same net amount of energy. They just enter the glycolysis pathway at different points, but still spend and gain the same amount of energy. Now that we've talked about the entire process of glycolysis, let's talk about normal energy yields for glycolysis. Under normal aerobic conditions, that's conditions with oxygen, we'd want all the reduced cofactors like NADH and FADH2 to be oxidized in the electron transport chain to give us energy. If that was the case, glycolysis would yield 2 ATP and 2 NADHs that then we could use to make 2 times 2.5 ATP. Remembering that that 2.5 ATP corresponds to the yield in ATP of NADH in the mitochondria. 2 ATP from glycolysis directly plus those 5 ATP that we could get if it was directly in the electron transport chain would normally yield a total of 7 ATP. The problem is we can't actually get the NADH into the mitochondria. To compensate, our bodies use dihydroxyacetone phosphate, which is much smaller and can move in and out of the outer mitochondrial membrane to accept electrons from NADH. The reduced form, glycerol 3-phosphate, can also move freely through the membrane and move electrons to a redox cofactor inside. The problem is that some energy is lost in this process, and now the electrons can't remake NADH, they only have enough energy to make FADH2. So the 2 NADH produced in the cytosol by glycolysis are really only worth 2 FADH2 once they're moved inside the mitochondria. Our total yield then becomes 2 ATP directly from glycolysis plus 3 ATP from the NADH in the cytosol once their electrons are transferred to FADH2 in the mitochondria for a total of 5 ATP from glycolysis, and then 2 pyruvate as well. The pyruvates that we produced in glycolysis can actually be further oxidized to produce even more energy. They can be oxidized to acetyl CoA and then processed in the citric acid cycle in the electron transport chain. That's actually the main way we can get energy out of glycolysis. But we need oxygen to do this, so this is only the pathway when we are in aerobic conditions. That's conditions with oxygen. When we're in anaerobic conditions, conditions without oxygen, we can't use the electron transport chain. This leads to a significant problem. The NADH that we'd like to use for energy can't be converted back to NAD+ to keep going through glycolysis. So in anaerobic conditions, again conditions without oxygen, we'll ferment the pyruvate, reducing it with NADH to restore the NAD+. This is less desirable by far. We can't get energy from the pyruvate or the NADH, but it's necessary because it's the only way to regain NAD+. The purpose of fermentation is to regain that NAD+ and allow us to get a small amount of energy from glycolysis. Anaerobic pathways allow us to get at least a little bit. Let's hone in on the differences between these fates of pyruvates a little bit more closely. The optimal route for pyruvate occurs in aerobic conditions. Pyruvate can be oxidized in the mitochondria to acetyl CoA. The acetyl CoA can then be used to make much more energy using the citric acid cycle and the electron transport chain. Each glucose will give us 2 pyruvates, so there also would be 2 NADHs produced by their oxidation to acetyl CoA, and 2 acetyl CoAs that can be metabolized in the citric acid cycle and the electron transport chain. The total yield for glucose being produced aerobically then is 5 ATP from glycolysis plus 2 times 2.5 ATP for the 2 NADHs produced by oxidizing pyruvate to acetyl CoA, and then 2 times 10 ATP for the acetyl CoAs that get processed in the citric acid cycle and the electron transport chain. This totals to 30 ATP, which is significantly larger than the energy of glycolysis alone. In contrast to the 30 ATP we get from oxidative pathways, the anaerobic pathways that humans use, reducing pyruvate to lactate, produce much less energy. The NADH we would have gotten energy from in glycolysis needs to be oxidized by the pyruvate instead, which eliminates any energy gain from the NADH and any energy gain from the pyruvate. We're left with only the ATP produced directly from glucose, a measly 2 ATP. Anaerobic conditions are much worse at giving us energy. So why do we go through anaerobic pathways? If a tissue is temporarily depleted of oxygen, we want it to temporarily function without oxygen, even if it's far less efficient. This is especially important for muscle tissue, where activity can quickly use up all the available oxygen. If you couldn't process any energy anaerobically, your muscles would seize. Or, if your brain ran out of oxygen, you'd stop being able to function if you couldn't do anything anaerobically. We make lactic acid, but yeast and some bacteria, and even goldfish, can make ethanol instead of lactic acid by reducing pyruvate. The goal for the organism isn't the ethanol, even if it is a brewer's goal. The organism makes the ethanol to allow it to restore the NAD+ that it needs for glycolysis, and allows it to continue to survive in temporarily anaerobic conditions. Notice that the end result is the same for the lactic acid route and for the ethanol route. We only get the ATPs directly produced from glycolysis. We don't get energy from any other source. Again, 3 fates are possible for pyruvate depending on the organism and conditions, but oxidizing pyruvate to acetyl CoA allows us to enter central metabolism and get much more energy than any of the anaerobic pathways. We've now talked about how we can use glucose, or fructose or galactose, to produce energy, either 30 ATP per hexose in aerobic conditions, or 2 ATP per hexose in anaerobic conditions. But what happens if we have enough ATP already? We certainly don't want to keep churning through glycolysis and using up all of our glucose. We want to store it for later. In case you forgot it from chapter 18, the major way that humans store energy as carbohydrates is the polysaccharide glycogen. So we need to be able to make that from glucose, and we need to be able to harvest the glucose later. When we make glycogen, the process is called glycogenesis. Notice that this name includes glycogen, which should be a giveaway that it involves glycogen. The name also includes the word genesis, which I hope many of us know means beginning. Many anabolic pathways that are building new large molecules from small molecules will include the word genesis. When we harvest glycogen, the pathway is called Glycogenolysis. Again, the word glycogen is part of the name, which should suggest to you that it involves glycogen. Lysis is also part of the name, which is common for catabolic pathways. For example, glycolysis. We'll talk about each of these pathways in more detail, starting with glycogenesis, the anabolic pathway that builds glycogen. Glycogenesis always occurs when we have more glucose than we need for energy. Cells will still pick up the glucose and phosphorylate it in the same way they would for glycolysis, so we can start from glucose 6-phosphate. We've already spent one ATP, but it's one that we would have spent for glycolysis anyway. The phosphate needs to be moved to make the hemiacetal more active for glycogenesis. The glucose 1-phosphate formed in the last step needs to be further activated, and to do that we use a uridine triphosphate, or a UTP. Because the UTP loses 2 phosphate pieces at once, this is the equivalent of losing 2 ATP. But now we have a much more active UDP glucose complex. The UDP activated glucose can bind with an existing glycogen chain to increase its length by 1. It costs 2 ATP per glucose to do this. Notice that this means that if glycogen is being consumed anaerobically, we get a net yield of 0 ATP. This doesn't matter because we're always synthesizing the glycogen when we have excess energy. The 2 ATP cost is negligible when we have plenty of energy. And the 2 ATP gain in anaerobic conditions is when we have no other options. Once we have glycogen, we can use it whenever our glucose levels drop low enough to necessitate releasing more. Glycogenolysis is the process by which we break down glycogen to make glucose when glucose levels are low. We're never going to have both glycogenesis and glycogenolysis active at the same time. They're reverse processes. We actually gain a phosphate on the glucose when we break off the glucose from glycogen, which is the equivalent of gaining an ATP. If the glucose is going to be used for glycolysis in the cell, that phosphate just needs to be moved back onto the 6th position, and then the glycolysis can continue. If the glucose is going to be used elsewhere, the phosphate is removed without energy gain, essentially losing an ATP. Again, we get slightly less energy back from the glycogen than we put in to make it, so glycogenesis and glycogenolysis will never be occurring at the same time. Again, it's important to see these pathways together so that we can see how they relate. Every pathway we've talked about, glycolysis, glycogenesis, and glycogenolysis, all start the same way. Glucose gets locked in the cell by phosphorylation. If there's lots of energy already, the glucose 6-phosphate will be converted to glycogen. If there isn't as much energy, the glucose 6-phosphate may be converted to energy in glycolysis. If glucose starts to drop low enough, glucose 6-phosphate can be made from glycogen in glycogenolysis and then either be used in glycolysis or exported to other tissues. We've talked about how we can break down glucose for energy and how glucose is able to produce energy anaerobically by fermenting pyruvate. In humans, pyruvate fermentation yields lactate, or lactic acid, the conjugate acid. The problem is, this lactate can't do anything for us in an anaerobic environment, and it just builds up, which in turn drops blood pH. Our bodies solve this by taking lactate from the anaerobic tissues, which are most likely muscles, and moving them into our liver. The liver tends not to go anaerobically as quickly as the muscles, and tends to use fats as an energy supply. The liver will convert lactate back to pyruvate, and then use the energy from fats to build pyruvates back into glucose. That glucose can be returned to the muscle tissues and anaerobically digested. The whole process, the Cori cycle, prevents lactate or lactic acid from accumulating to the extent that they otherwise might, and allows us to keep a steady supply of glucose while we're exercising. But this process requires a reverse of glycolysis, where pyruvate can be converted back into glucose. That process is called gluconeogenesis. Gluco for glucose, neo for new, and genesis for beginning. Glucose new beginning, gluconeogenesis. Again, notice that the anabolic pathway here has genesis as part of the name. Gluconeogenesis is in many ways the reverse of glycolysis, but not exactly. The jump from phosphoenol pyruvate to pyruvate is so favorable that we can't go directly back the same way. We need to create oxaloacetate first, and then phosphoenol pyruvate. This costs way more energy than we get from glucose. Notice it costs 6 triphosphates, 4 ATP and 2 GTP, while we only get 2 ATP going the other way. We'll always be going through gluconeogenesis in a different tissue from glycolysis, and it will always be powered by catabolism of a different type of molecule, typically fats. If we don't have these separated, we'd run into a problem we call a futile cycle. We keep on going from glucose to pyruvate and then back, but we'd lose energy every time. We've now talked about 4 pathways relating to carbohydrate metabolism. They all start with G and are all very similar. I'm sorry, I didn't name them. It's still really important to keep these straight. 3 things that may be helpful. 1. Every catabolic pathway has lysis as part of its name. Remember that these are the pathways that go from large to small. Every anabolic pathway has genesis as part of its name. Remember that anabolic is small to large. And 3. Every pathway that involves glycogen has glycogen as part of its name. This can help at least a little bit. In addition to these 4 pathways, note that we also have several possible routes for pyruvate. Anaerobic to squeeze out a little bit of energy from glucose if we don't have oxygen, or aerobic to get way more energy from the citric acid cycle and the electron transport chain. There is a 5th pathway relating to glucose, the pentose phosphate pathway. The pentose phosphate pathway is a catabolic pathway, similar to glycolysis. However, glycolysis is being used to supply energy either anaerobically or aerobically in the form of ATP. The pentose phosphate pathway specifically gives us energy in the form of a reduced cofactor NADPH. The structure of NADPH is shown on the left. The only physical difference between NADH and NADPH is that NADPH has a phosphate on the 2' position of the ribose. This allows different enzymes to recognize and use NADPH than NADH. NADPH is used specifically for synthesizing fatty acids. We'll talk more about fatty acid synthesis, also called lipogenesis, genesis for anabolic pathways, in the next chapter. This figure shows a visual summary of what we've talked about in this chapter. When you ingest carbohydrates, they are digested to monosaccharides, mostly glucose. Glucose can be used for energy or stored for later. When glucose is used for energy, it typically goes through glycolysis to pyruvate, which can be metabolized aerobically in our central metabolism or anaerobically for far less energy. It can also be catabolized through the pentose phosphate pathway to get NADPH specifically to power fatty acid production. If those 2 pathways are active in a cell, glycolysis and the pentose phosphate pathway, we probably have more than enough glucose. We will probably also try to store some for later as glycogen through glycogenesis. On the other hand, if glucose is low, we may break down glycogen through glycogenolysis, and in the liver, we may synthesize glucose from pyruvate with gluconeogenesis. There's quite a bit of logic behind when these pathways are working. When we need energy, the pathways that give us energy are likely to be active. When we need glucose, the pathways that give us glucose are likely to be active. When we need to get rid of glucose, the pathways that use glucose are likely to be active. All of this lays an important foundation for understanding hormonal control of metabolism. We have 3 major hormones that regulate our metabolism in really broad ways: insulin, glucagon, and epinephrine, which is essentially the same as adrenaline. Insulin is the hormone that says there's too much glucose, so it activates everything that uses up glucose, glycolysis and the pentose phosphate pathway as catabolic pathways, but also glycogenesis, making glycogen as an anabolic pathway. It will cause everything that makes glucose, like glycogenolysis and gluconeogenesis, to stop. Glucagon is the opposite of insulin. Instead of triggering the use of glucose because there's too much, glucagon signals glucose is gone and triggers the retention and synthesis of glucose because there's too little. It will slow down glycolysis, prevent glycogen synthesis by glycogenesis, and prevent the pentose phosphate pathway. It will also activate glycogenolysis. It also activates gluconeogenesis to make more glucose if muscle tissue is anaerobic. Epinephrine or adrenaline is a little bit different than those. Epinephrine is a way of triggering a sudden burst of energy. Think an EpiPen. Only catabolic pathways are activated, and pretty much all the catabolic pathways are activated. Everything with a lysis. So glycogenolysis is going to give us glucose for glycolysis. And we'll also see later that epinephrine will activate catabolic pathways for fats too. Again, anything to give us that burst of energy for a fight or flight response.